And finally, we come to transfer RNA. So we talked about messenger RNA. We talked about ribosomal RNA briefly. Now we're going to talk about transfer RNA. Transfer RNA, you can see that it has this clover leaf-like structure. There's 3 leaves. It must not be a very lucky clover. It's just 3 leaves to the clover. And of course, at the 5' end is a phosphate group going all the way around to the 3' end where there's a hydroxyl group. Now, in transfer RNA, it's to this 3' hydroxyl group to which the activated amino acid is going to be attached. You see, the function of transfer RNA are twofold. Number one, to pick up the amino acid, to pick up the proper amino acid, and then number 2, to recognize the codon on the messenger RNA. So there's 2 important parts to each transfer RNA. Number 1 is that 3' hydroxyl group where the amino acid is going to be covalently attached. Notice it's always a CCA residue here. There's always a CCA residue to which the incoming amino acid is going to covalently bond to the transfer RNA. The second important portion of each and every transfer RNA is the anticodon. Okay, the anticodon down here is the sequence that's going to be complementary and antiparallel to the codon sequence of the messenger RNA. For example, if the incoming messenger RNA places an AUG in the proper site on the ribosome, well, that AUG is going to want to complementarily base pair with a transfer RNA that is antiparallel and complementary to it. So how would that read? Well, from 5' to 3', that's going to read CAU. The C complementary to the G, the A complementary to the U, and of course, the U complementary to the A. So that's how this would work. It's complementary, remember, once again, and it's antiparallel. So if the codon reads from 5' to 3', such as this, it reads AUG, the anticodon from 5' to 3' is going to read CAU. You might be asked a question where you're given the mRNA sequence and asked, what would the anticodon look like? Or you may be even given a DNA sequence and have to work your way down and ask and figure out what would the anticodon on the tRNA look like. Now, just a couple other key features. Number 1, transfer RNA is unusual in that you can see modified bases. Like you see, the guanosines can be modified. The adenosines can be modified. And lo and behold, what do you see down here? You see a T. Oh my goodness. T, this is RNA. You're not supposed to see T's in RNA, are you? Well, it's okay because in transfer, tRNA, sometimes you see T's. So remember that. That'll help you remember the fact that sometimes in transfer RNA, you can see the T's. The letter's right there for you. The other point I wanted to emphasize is there's a special family of enzymes that are responsible for covalently attaching the amino acid to the 3' end of the transfer RNA. What are these families of enzymes called? Well, they're called amino-acyl tRNA synthetases. So the name tells you exactly what the enzyme does. It's an amino-acyl tRNA synthetase. So through an acyl linkage, amino groups of the amino acid are attached to the tRNA. So every single, depending upon the anticodon that's present on the transfer RNA, the anticodon is going to get the special amino acid attached to it, which would then place the proper amino acid in the protein when the anticodon base pairs with the codon of the messenger RNA. So that's how transfer RNA works. We'll look at the process of translation a little bit later itself, and that will make a lot more sense to you. So just to emphasize some important points about transcription and RNA processing in general. First of all, between prokaryotes and eukaryotes, gene regions. In prokaryotes, they may be polycistronic, whereas in eukaryotes, they're always monocistronic, always going to be monocistronic. In prokaryotic genes, the genes are always continuous coding regions, whereas in eukaryotes, the regions can be divided into exons and then the interrupting intron portion. There's very little spacer or non-coding DNA in the prokaryotic genome, whereas for eukaryotes, there's going to be lots of spacer DNA, lots of non-coding DNA regions. As far as the RNA polymerases go, in prokaryotes, the RNA polymerase, the core enzyme, it's an alpha-2 beta prime. That's the core enzyme. Alpha-2 beta prime is what takes the prokaryotic DNA and transcribes it into messenger RNA, or it makes the ribosomal RNA, or it makes the transfer RNA. In eukaryotes, we have a different story. We have at least 3 distinct RNA polymerases. You might be asked about these to differentiate them. RNA polymerase 1 makes ribosomal RNA. RNA polymerase 2 makes the messenger RNA and also the small nuclear RNA involved in splicing. RNA polymerase 3, well, it makes transfer RNA and that 5S RNA. Remember that little exception. It makes the 5S ribosomal RNA. How is transcription initiated? In prokaryotes, you have a promoter region with a TATA box. You also have a -35 sequence. We didn't discuss too much because it's not that important. But you have a -10 TATA box, a -35 sequence, which are the landing lights which the prokaryotic RNA polymerase looks for. There may or may not be a sigma factor utilized to initiate transcription in prokaryotes, depending on the particular gene. In eukaryotes, the TATA box is located at the -25 sequence, so it's a little further upstream. And there's also this cat box at the -70 position. The cat box, also known as the El Gato box to my Spanish-speaking friends, the cat box is simply a promoter region where the RNA polymerase can look for and see the proper sequence, bind, and then start transcribing. In addition, in eukaryotes, there's transcription factors such as 2D, transcription factor 2D, which can bind to the promoter and help elicit initiation of transcription. As far as the mRNA synthesis goes, notice this is the same for both prokaryotes and eukaryotes. The template is red, 3 to 5 prime. The mRNA is synthesized 5 to 3 prime. That's never going to change. Transcription is going to begin at the +1 base. Remember, there's no 0 base. It begins at +1. So we move on to how transcription is terminated. In prokaryotes, there tends to be a stem loop sequence, maybe a poly-U region. There may be a stem loop sequence and a row factor. It just depends. In eukaryotes, this is not well characterized. So one less thing for you to remember. That's good. How is the DNA transcript related to the RNA? Once again, for both prokaryotes and eukaryotes, RNA is anti-parallel and complementary to the DNA sequence. Remember that the messenger RNA is going to be identical to the coding strand, except U is going to be substituted for T's, because after all, there's no U's in the DNA and there's no T's in the RNA, except for, well, we just mentioned, in transfer RNA, sometimes you will see T's in transfer RNA. And lastly, processing. So processing of the heterogeneous nuclear RNA or the pre-mRNA, there's absolutely no processing that's going to go on within prokaryotes, whereas with eukaryotes, in the nucleus, there's a 5' cap, so there's a 7-methylguanosine cap that's going to attach to the 5' end. There's a poly-A tail, a polyadenosine tail, attached to the 3' end of each message, and then there's going to be removal of the introns. So there's going to be splicing of the introns that occurs, and so the exons are then linked together after the introns are removed. And sometimes there can be alternative splicing that happens between various cell types. A big difference here is going to be the ribosomes. Once again, in prokaryotes, it's a 70S complete ribosome divided into a 30S and 50S small and large ribosomal subunit. There's other ribosomal RNA species in there, and there's proteins. In eukaryotes, there's a complete 80S ribosome, which is subdivided into a small 40S and a large 60S ribosomal RNA. This ribosomal RNA is going to bind to various proteins, which gives the ribosome its 3-dimensional structure. In both prokaryotes and eukaryotes, the structure of the tRNA is relatively the same. It's that clover-leafed structure attached to the 3' end via the amino acyl tRNA synthetases is going to be a covalent attachment of an amino acid, and then of course there's going to be an anticodon present, which will complementarily base pair with the appropriate messenger RNA codon. So that concludes Chapter 3, in which we discussed transcription, RNA processing. The key points here, once again, are going to be recognizing the difference between prokaryotic and eukaryotic transcription, processing events, etc. Lots of questions on the exam are going to make sure that you're clear as to what's going on in prokaryotes and what's going on in eukaryotes.